Catherine Talbot is an assistant professor in the School of Psychology at Florida Tech, co-director of the Animal Cognitive Research Center at Brevard Zoo, and developer of the Florida Tech's new Animal Behavior and Cognitive Bachelor of Science program, which launched this year. Her research explores the evolutionary and biological basis of sociality in non-human primates, focusing on topics such as face recognition, social cognition, autism, prosocial behavior, and behavioral inequality. Currently, Dr. Talbot and her collaborators are examining the relationship between biological markers of social behavior and performance on social cognitive tasks with direct relevance to autism. We're looking forward to hearing where her research is leading and what it can teach us next. So please help me in welcoming Catherine to the CFS.
Well, thank you for that introduction, and thanks for having me here today. So I am just going to go ahead and get started and talk to you about what monkeys can teach us about autism. So to give you a brief overview here, I'm going to be talking about social functioning and how it's related to autism, and I'll talk a little bit about animal models and provide you with an overview of our research program before talking about how we can screen for this sort of low social phenotype and what we've done to validate the measure that we developed and what we've learned about that low social phenotype and how we can translate that to humans with relation to biomarkers and potential therapeutic treatments. So autism is an early onset neurodevelopmental condition that's characterized by persistent social communication and interaction difficulties. And these difficulties span basic social information processing skills like face recognition and responding to other social communications cues. And these deficits really represent the extreme of a continuous distribution, since autistic traits are common throughout the subclinical population as well. It is particularly prevalent in males, such that 4 males are diagnosed to every 1 female. And 25 years ago, the Center for Disease Control and Prevention began systematically tracking the prevalence of autism across several sites in the United States. In 2000, 1 in 150 children were diagnosed with autism. And the report that was released this past spring, which is based on 2022 data here, reveals a very steady rise, such that 1 in 31 US children are now diagnosed with autism spectrum disorder. That marks a staggering 384% increase since 2000. Despite its prevalence, its disease mechanisms remain poorly understood. There are no lab-based tests. It's diagnosed behaviorally. There are 2 FDA-approved drugs, but they are antipsychotics, such that they treat associated symptoms like irritability, aggression, and self-injurious behavior. But there are no medications that target the core social deficits of autism. And you can imagine that studying disease biology directly in patients is incredibly difficult, impeding progress. And while much of the research has relied on rodent models, the evolutionary distance between rodents and humans is around 87 million years, and that presents significant differences in biological and behavioral functioning, right? In particular, mice lack the social cognitive abilities that are critical to modeling the core social deficits of autism. So some of those deficits include things like lack of eye gaze, poor facial recognition, poor theory of mind, so understanding others' perspective, and so forth. Rhesus macaques, these are rhesus macaque monkeys, they share 93% of their DNA with humans. And like humans, they live in complex social groups and exhibit stable and pronounced individual differences in social functioning. So that naturally occurring variation in rhesus monkey social behavior is a useful model for investigating the social impairments relevant to ASD. They also exhibit complex cognitive abilities that, again, are critical for modeling those core social deficits. So to give you an overview of our research program, we begin by observing the monkeys in these habitats, and this is at the California National Primate Research Center at UC Davis. And they're housed with anywhere between 50 and 150 individuals across various ages and sex, and that's really similar to what you see in the wild. And then we use data on their social behavior to classify them along the social spectrum. So do they spend a lot of time in the company of others, or do they spend a lot of time alone, and a few other simple measures that we look at. And then we identify the behavioral extremes, and then we sample those extremes, examining potential biomarkers, and test them on a battery of various cognitive tasks that are both social and non-social in nature. And so then we put all those pieces together to evaluate how natural social behavior, biology, and cognitive performance relate to one another. However, because social impairments associated with ASD reflect the extreme of a continuous distribution of social traits, researchers need a tool that can rapidly and accurately identify these extremes in the general population. So in humans, variation in social functioning has been measured using the Social Responsiveness Scale, which is a 65-item questionnaire about a person's natural behavior in social situations, and is typically filled out by children's caretakers and/or their teachers. And the Human Social Responsiveness Scale, SRS for short, was developed by a team of scientists that were the human SRS was adapted for use in chimpanzees by removing items that were associated with language. And this was done by a team of scientists that were based at the Yerkes National Primate Research Center. And the macaque version was developed as well from that. And there still needed to be some work done with that particular study. So in that study, they looked at a sample of 105 individuals, but 87 of them were female, and no young or socially impaired animals were included in this particular study. Their inter-rater reliabilities were done on 16 individuals, and they didn't really look at retest reliability. They also didn't compare their scores on the macaque version to any sort of social behavior. So our team did a lot of work to refine this macaque social responsiveness scale, because careful psychometric analysis of ratings is fairly rare, but that work is really necessary to evaluate the validity of the scale and the animal model itself. So this is all a bit technical and boring, so I've really summarized a few good years of my life in this one slide, but I will just provide you with a brief overview of all the work that we did with this. We optimized that scale by changing the scale itself. So we changed it from a 4-point scale to a 7-point scale to sort of accommodate a neutral response. We removed unclear terminology, especially when it comes to primates that were related to like emotion, because that's difficult to assess in an animal. And then we also expanded the species-specific descriptions of behavior so that it was very specific to the macaque monkeys. Then we refined the scale. And we refined the scales by conducting both inter-rater reliabilities and retest reliabilities on 116 monkeys. And that population that we did that with, it included 31 males and 85 females that were between 1 to 24 years old. Then in a second cohort of monkeys, it was 233 juvenile young males, so ages 1 to 7, we psychometrically assessed the scale. And to do that, we looked at the internal consistency reliability measures. We also looked at whether we had convergent construct validity, which just means that we were seeing how this related to their actual social behavior. And then we evaluated its predictive validity in terms of whether it predicted behavior in biology. And so I'm going to talk to you a little bit more about that because it's a little bit more interesting than the psychometrics. So when all was said and done, the revised scale consisted of 17 reliable items. And that word cloud there, you can see the descriptors of each of those items. And for instance, the full description of alone is would rather be alone with others. Bizarre, that description was behaves in a way that seems strange for others of comparable age, sex, or rank. And then fidgety seems more fidgety in social situations. And we know that age and rank can impact social behavior in non-human primates, so therefore we evaluated whether there was any effect of age or rank on macaque social responsiveness scale revised scores. So we found that there is no relation between MSRS scores and age in our sample here, and what you see here is why these are all clustered is just because rhesus macaques breed in, they have seasonal breeding, and so they're all born around the same time. So there's no relation with rank, with age, and there's no relation with rank. All right, and that really supports the idea that we are in fact measuring intrinsic social traits as intended, so we can't just explain this away by differences in some other measure. And then we use behavioral data to classify our subjects as either high social or low social based on the frequency of time they spent alone from those social observations. And we differentiated these by 1.5 standard deviations above and below the mean, respectively. And we chose to use 1.5 standard deviations because when parents and classroom teachers rate the severity of social deficits, the point of greatest differentiation from the respective distributions occurs at that point that's approximately 1.5 standard deviations from the population mean. So we use that based on the translational basis for that. And in the end, we classified 14 individuals that were high social and 14 individuals that were low social. So once we had classified the social extremes, we next asked whether these MSRS scores could predict those social groups. And so it's a bit counterintuitive here, so I just want to point out that higher scores on this rating scale are indicative of greater social impairment, and lower scores indicate more social adeptness. And so what you're looking at in this graph here, and I'm going to apologize that my talk is a bit data heavy, so if there's anything that you need clarification on, please just raise your hand and ask. But what you're looking at here is essentially a logistic regression, and you're looking at the probability of being low social based on the MSRS score, and low social individuals are represented by blue and high social and orange, and this is the 50% chance of probability. And so we classified those social extremes with 96% accuracy, where they correctly classified 27 out of the 28 monkeys. All right, and then we also, as would be expected, the converse was also true, and so you can see, and this is essentially a bar graph with the low social monkeys and the high social monkeys represented there, and low social monkeys exhibit greater social impairments compared to the high social monkeys. So next, to better understand the psychological factors that contribute to variation in social functioning, we evaluated the underlying factor structure of this rating scale. And we used an exploratory factor analysis to do this. And an exploratory factor analysis looks at underlying latent constructs or factors that explain the shared variance among observed variables. So it assumes some unobserved variables cause the correlations that among the measured items. So using this analysis, we found 3 factors that explain over 68% of the variation. And 2 of those factors were related to sociability. And so I'm going to focus mostly in the next couple slides on this poor social motivation and poor social attractiveness, which is what we label them. So that also suggests that the scale is measuring what it theoretically should be, right? It's measuring one's ability to function socially. And what you're looking at here is essentially the items from the MSRS-R that loaded onto these factors. And a positive loading is a 0.4 or greater. And then whether it's negative or positive represents the direction in which it loads. So I've only included those that were significant here on each of these factors. And the sort of traits that are associated with those. And so the factor 1 had 10 items that were related to social avoidance or poor social motivation. And actually 7 out of 10 of these items actually map to the human SRS social motivation subscale. And factor 2 included 3 items, all of which related to some sort of disruptive behavior. And all 3 of those items actually map to the human SRS of repetitive mannerisms. And then factor 3 items, there's 4 items related to poor social attractiveness, and all 4 of those items map to the human SRS social communication subscale. And so next, we evaluated whether the resulting MSRS-R factors predicted quantitative social behavior measures. So both grooming and what we found is that basically poor social motivation was associated with increased time spent alone, less contact with other monkeys, and less grooming behavior. And so grooming in non-human primates is essentially when they pick through the fur of another individual, and it does help reduce parasites, but it also is really important for social bonding in primates. And so there is a negative association between poor social motivation and grooming there. And then poor social attractiveness was associated with increased grooming and a decrease in play. And so both grooming and play behavior function in the formation and maintenance of social bonds in primates, suggesting that those individuals that are not socially attractive partners and perhaps not well coordinated or communicative may employ a different social strategy and spend more time grooming and less time playing with their more socially attractive peers. And just in case you were interested, inappropriate behavior didn't have any, that factor didn't have any relationship with the social behavior that we measured through our observations. And so then we evaluated the relationship between these factors and macaque personality dimensions. And so we found that poor social motivation, MSRS-R factor, was associated with less sociability, less confidence, and less irritability. Whereas the poor social attractiveness was associated with less sociability, but higher confidence and irritability. And so it is possible that what makes an animal socially unattractive is an overconfident and irritable personality, which together may result in less social opportunity. And so I sort of think of this as like the annoying guy at the bar at the end of the night, right? We also, given that low social monkeys, right, show diminished capacity to respond to species-typical behavioral cues. And so it's maybe not that surprising that these MSRS scores also positively predicted rates of trauma and total number of traumatic injuries, such that the more socially impaired monkeys experienced more trauma compared to high social monkeys. So this is really similar to, right, low sociality is sort of linked to peer bullying, right? And in this case, it held true even after controlling for rank, because these macaques do have a very strict dominance hierarchy, and so we do want to make sure that we're controlling for rank in that sense. And so it's pretty impressive that this is impacting their injury rates after controlling for that. So we next turn to look at potential biomarkers of social functioning, and 2 candidates are oxytocin and vasopressin. So have you heard of these two hormones before? Raise your hand if you have. Yes? Okay, great. So there are two structurally and functionally similar hormones that are known to regulate social functioning in mammals, not just humans, but really conserved across mammals. And oxytocin is best known for its role in female reproduction. Right? It's produced in large amounts during childbirth and with stimulation of the nipples during nursing. It's released during orgasm in both males and females. And more recent studies suggest that it increases social recognition and bonding. It has anxiolytic effects such that it reduces anxiety, and it has been linked to trust in humans. And so vasopressin is very similar, but we know less about it. There is a suggestion that oxytocin is responsible for maternal behavior, whereas male typical social behavior is associated with vasopressin, although I think that is a somewhat limited perspective. Vasopressin has been suggested to play a role in some cognitive abilities, including social memory, as well as emotionality, and it has been associated with social withdrawal. So we really focused our search for biomarkers on these two neurohormones, although we also looked at other potential candidates and found that the MSRS-R score negatively predicts vasopressin in cerebral spinal fluid. That cerebral spinal fluid is a fluid that surrounds your brain. And so individuals with the lowest CSF vasopressin concentration demonstrated the greatest social impairment. So you can see here that you've got the concentration down here, and then those with the lowest concentration display the highest, greatest social impairments on the MSRS score. So to summarize what we've discussed thus far, the MSRS-R measures what it's supposed to, right? And it's also predictive of behavior and biology, meaning that it can serve as a high throughput screening tool for scientists to quickly and accurately identify socially impaired individuals in non-human primate populations. We have also learned more about the low social phenotype in our monkey model. For instance, we know that social functioning is stable across time, and I'm sorry for this wall of text here, but what essentially this is the study that we looked at different social behavior across several years from 2016 to 2017. And this is just sort of across 3 different studies where we looked at, we classified the individuals, we looked at group differences, and then we looked at the behavioral stability of their phenotype over time, so across those 2 years. And what we found here is sort of the study 1 conducted in 2016. The low social and high social individuals remained the same as to 2018. So in this case, we had a very large sample, but we only had 11 monkeys at this point in time that we could sample. But all of them remained consistent to their social classification. And we found that low sociality was linked to the initiation, but not the receipt of prosocial behavior. And when I say prosocial behavior, I'm really referring to the acts that benefit the welfare of others. Okay? And so we have low social monkeys in blue here and high social monkeys in orange. And what we see is that they initiate significantly less prosocial behavior compared to high social monkeys. But there's no difference in the receipt of prosocial behavior. And then we found that was actually specific to prosocial behavior because they did not have any differences in terms of initiating threats and receiving threats. So it was really sort of specific to the prosocial domain, this difference here. Low social functioning is, we found that low social functioning is paternally but not maternally derived. So that supports a paternal origin effect. So normally genes are expressed the same way whether they come from the mother or the father. But with parent of origin effects, the same allele can have different effects depending on whether it was inherited from the mother or the father. And so the ideal strategy for determining these effects is by using half-sibling analysis, where half-siblings share one but not both parents, and each individual belongs to a unique combination of paternal and maternal half-siblings. So this family structure is fairly rare in humans, but rhesus macaques have a promiscuous breeding system that consistently produces paternal and maternal half-siblings. Again, just emphasizing the importance of animal models. And so this graph here is showing the genetic contribution of the parent, and it's presenting the best linear unbiased predictors, is what each dot represents. So basically, each dot represents a half-sibling group, a set of sons who share the same father in the blue dots, or the same mother in the orange dots. So on the y-axis, you have the genetic contribution to sons mean score. Basically, that means how much of the parent's genes seem to push their sons above or below the average for social behavior, right? And the average is standardized at 0 here. And so this is in terms of, this is the non-social, it's an equivalent score, so we standardize the score. And we can see that fathers really contribute to the variation in whether they're offspring or very high, highly social or low social. And that is also true for, we found the same sort of relationship for the MSRS score. And this is, remember that higher scores mean greater social impairment. So what you're actually looking at here is those individuals that were more social are actually down here. So this data point is actually sort of this one down here. And so we found essentially that the father's genetics makeup explains a lot of variation between sons. So now we are going to talk a little bit like how do we take this information and translate it to humans, right? So our hope is really to better understand the biological signaling pathways underlying naturally occurring social functioning, as they can reveal promising molecules for ASD therapeutic development. And hopefully this can help accelerate the discovery of potential biomarkers and streamline the development of effective therapeutics. So we know arginine, the full name is arginine vasopressin. It serves as we know that it serves as a biomarker for social functioning in our monkey model. And in this case here, you're looking at a very similar graph to what I've shown you before, that low social monkeys are depicted in the blue circles there, and high social monkeys in the orange circles. And we're looking at the probability of being low social along with their CSF vasopressin concentration. Okay, and essentially this CSF vasopressin concentration predicts low sociality with 93% accuracy. And this is true that there is a significant difference between low and high social monkeys as well in their CSF vasopressin concentrations. We then translated that to a small cohort of children with autism, and we found that it also predicted children with autism with 93% accuracy here. So this is the probability of being diagnosed, and then you've got their vasopressin concentration here. There is one mislabeled here. So vasopressin is a neural hormone that originates in the hypothalamic pituitary gland, and when our body produces it naturally. And so you can find it we'll talk a little bit about this, but you can find it in your blood levels. You can also find it in your cerebrospinal fluid levels. And so it is, been implicated like oxytocin in social bonding. And for instance, we don't have as much data on vasopressin, but oxytocin, if you look at your dog in the eyes, actually you both experience an increase of oxytocin. And so it's really associated with social bonding and those sorts of things. So then we next sought to test, so we found that this is a potential biomarkers in our monkey model, and that it's translated to a small patient cohort of autistic patients. And then we wanted to test it as a potential therapeutic. So we sought to test the effect and specificity of vasopressin administration on social and non-social cognitive processes in previously identified low social monkeys. And so we know that low sociality is correlated with a high autistic-like trait burden, and this was true in this particular sample as well. So we first developed a method of drug administration in which monkeys could voluntarily participate. So that helps to reduce any confounding effects of stress, and it allows for sort of more immediate translational potential to pediatric clinical populations. And so we use this method of administration in 2 different studies where monkeys received either 0, which is just our placebo, was a vasopressin-specific placebo, meaning it had all the same things except for the vasopressin itself. Either that or 25 IU vasopressin or 50 IU. And IU just means international units. And so it's just the amount of vasopressin in that particular dose. And we then looked at, in study 1, the behavioral effects of it using on a object recognition task, a social competence task that was displaying another monkey that was either displaying affiliative behavior, so friendly behavior, or aggressive behavior, as well as a face recognition task. In study 2, we evaluated whether we saw an increase in vasopressin in cerebral spinal fluid as a result of this method of administration. So we just wanted to validate what we were doing and making sure that it actually was increasing levels when we administered it. However, endogenous vasopressin release and central vasopressin administration have both been shown to induce social bonds and parental care in a variety of male rodents. However, vasopressin has also been shown to induce agonistic behavior in males under certain circumstances. So it increases aggressive behavior. All right, so one important limitation of this, though, is that much of this research was conducted in neurotypical animals, limiting its translational impact for autism. So we really wanted to assess vasopressin's effect in socially impaired animals to be able to really evaluate its ability to sort of rescue these species-typical social functioning. But we wanted to also make sure that it was not increasing aggression, right? Because that would not be good for the translational value. We don't want to increase aggression in autistic patients. So we had several hypotheses. One, that vasopressin, but not placebo, would improve memory for faces. 2, that vasopressin would not alter object recognition performance. We were hoping that it would be specific to the social domain. Right? And that vasopressin would enhance social responsiveness to friendly signals, but not aggressive signals. And finally, in study 2, we evaluated, it's really called the pharmacokinetics of vasopressin administration, and expected that it would increase concentrations in cisternal, cerebral spinal fluid, and plasma as well, so in blood. So what we see here in these graphs I'm going to show you in the next few slides, the 25 IU dose and the 50 IU dose are plotted together. And what you're looking at here is the ratio of looks to the novel versus familiar stimuli. So basically what this test does, both of these, this object recognition and memory, face recognition memory, present to individuals for some period of time, and then so that you become familiar with those faces or objects, and then it presents you with the same, the old face or object and a new one, right? Humans have it, and non-human primates have an innate preference for novel objects and stimuli, and so that's essentially what we're testing here. Could they remember what they had seen previously, and did they have differences based on objects, whether it was social or non-social stimuli? And so you have the looks to novel versus familiar ratio here, plotted by the treatment. So we've got placebo, and again, those 2 doses are plotted together. Blue dots are represented by the faces task, and then this is their performance on the object task. And so let's just look at placebo first. So essentially low social monkeys perform much better at object recognition memory compared to face recognition memory. This is true of autistic people as well. They have a preference for inanimate objects. When administered vasopressin, their performance on the face recognition memory task was essentially rescued up to the level of the performance on the object recognition task. So it was specific to the social domain. Then we were looking at whether how they responded to species-typical communication cues. And so here again, we've got essentially affiliative responses to affiliative overtures by the stimulus monkey. So this is essentially a recording of a monkey displaying friendly behaviors that's presented to our subjects. In this case, it was displaying aggressive behaviors. And so what we're looking at here is the affiliative responding to social versus non-social stimuli, because these bouts of video were interspersed with non-social behavior as well. And so we've got affiliative responding here in blue and aggressive responses in orange. And so on placebo, there is no difference in friendly versus aggressive responding from these low social monkeys. But when they were administered vasopressin, they showed an increase, a significant increase in that friendly responses to friendly signals from the stimulus monkey. So it's increasing their prosocial behavior. Then in this next box here, we've got the same thing to aggression. So we've got aggressive responses to social versus those non-social video blocks. And you can see in placebo there is no difference here. And then again when they were administered vasopressin, there's still really, this is not significant. There's no difference in aggressive responding to aggressive stimuli. We next looked at cerebrospinal fluid and plasma, looking at the vasopressin levels there. And here what we are looking at is all data are expressed as the ratio of vasopressin concentration to placebo at each time point that we measured it following administration. And this sort of helps just control for a number of things. So placebo served as a control for any sort of vehicle effects, any circadian effects, because we see circadian effects with many hormones, including vasopressin. Although we did also test them at the same exact time each day, it also controlled for any sort of systematic osmolarity effects of fluid administration. So in that first panel there, you're looking at levels in cerebrospinal fluid, and the 25 IU, the lower dose is plotted in blue and the higher dose is plotted in orange. And so we observed a progressive linear increase in the CSF vasopressin concentration over the course of the 2 hours following administration. And in plasma, levels also changed significantly following nebulization. And we found this sort of quadratic rise and fall in vasopressin levels. So for both of these, for both CSF and plasma, these effects were consistently stronger for the larger dose than the 25 IU dose. Okay, so we did find that vasopressin selectively improved performance on social tasks. On placebos, low social monkeys prefer to look at objects more than faces, similar to people diagnosed with autism, and when administering vasopressin, it did not alter their performance on object recognition memory. And then it also enhanced social responsiveness to affiliative signals. And so it was increasing prosocial responses while it did not induce aggression, even when we were trying to elicit it. And finally, that we confirmed that our method of vasopressin nebulization penetrated both the cerebrospinal fluid and blood, albeit with a bit different time courses. And so these findings suggest that this sort of voluntary vasopressin nebulization does penetrate the central nervous system, and it rescues species typical responsive to social information in low social animals without inducing aggression. Yay. So, it would be a great, I didn't put a slide on here for future directions, but I figured you would probably have a lot of questions about that. One of the things, I'll just mention a couple that come to mind. We want to this sort of paves the way for studying at-risk monkeys, infant monkeys, so younger monkeys, to examine whether if we can administer vasopressin at a younger age, can it help change the developmental trajectory of their social functioning. Because, when you look at the mechanisms by which these sort of motivate behavior, it might be that once you start getting that sort of spike in, sort of reinforcement for paying attention to social things internally, it might help you essentially create that positive feedback loop of appropriately responding to social information. Thank you.